Good morning. It's January 3rd, and this is your daily brief in science. Here's everything you need to know. A new study from Karolinska Institutet in Sweden reveals that artificial intelligence can surpass human experts in detecting ovarian cancer using ultrasound images. Published in Nature Medicine, this research highlights the potential of AI in enhancing diagnostic accuracy in healthcare. The study utilized a robust dataset of over 17,000 ultrasound images from 3,652 patients collected across 20 hospitals in 8 countries. The AI models demonstrated an accuracy rate of 86.3%, outperforming human experts who achieved an accuracy of 82.6% and non-expert examiners at 77.7%. Professor Elizabeth Epstein noted the global shortage of ultrasound experts, which can lead to misdiagnoses and unnecessary procedures. The AI's ability to assist in challenging cases and in areas with limited expert availability could significantly ease the strain on healthcare systems. Philip Christiansen, a doctoral student and co-author of the study, stated that adaptable AI tools could optimize healthcare resources and reduce expert workloads. However, researchers emphasize the need for further studies to explore the clinical limitations and full potential of these neural network models in diagnosing ovarian cancer. Current clinical studies at Södersjukhuset are assessing the safety and effectiveness of the AI tool in real-world settings, with plans for a randomized multicenter study. In simulated triage scenarios, the AI support reduced the need for expert referrals by 63% and lowered the misdiagnosis rate by 18%, improving patient care efficiency. Additionally, Epstein and her co-authors have applied for a patent for their diagnostic methods through their company, Intelligen. The study was conducted in collaboration with researchers from KTH Royal Institute of Technology and received funding from various Swedish research grants. It's important to note that the content has not been reviewed by the American Society of Clinical Oncology, Inc., and does not necessarily reflect ASCO's viewpoints. NASA's Parker Solar Probe has set a new record by flying within just 3.8 million miles of the Sun's surface on December 24, 2024. This historic milestone surpasses its previous record of 7.26 million kilometers achieved in 2023. During this close approach, the probe reached astonishing speeds of approximately 692,000 kilometers per hour, making it the fastest human-made object ever recorded. After a period of silence as it navigated the Sun's atmosphere, the probe confirmed its successful flyby by sending a beacon signal on December 26, 2024. Telemetry data received on January 1, 2025, confirmed that all systems and scientific instruments were operational, allowing the probe to collect valuable data during this unprecedented journey. Looking ahead, the Parker Solar Probe is scheduled for 2 more close approaches to the Sun on March 22 and June 19, 2025. Scientists are eager to analyze the data collected during this flyby to explore mysteries surrounding the Sun, particularly why the corona is significantly hotter than the Sun's surface. The probe has already made unexpected discoveries about the outer corona's structure and the origins of solar wind switchbacks. In addition to its primary mission, the Parker Solar Probe enhances our understanding of space weather by documenting solar interactions with planets and capturing images of Venus during its gravity assists. NASA's Associate Administrator Nikki Fox highlighted that this mission is a historic moment for understanding solar impacts on the solar system and beyond. To endure the extreme temperatures of the corona, the probe is equipped with a 4.5-inch thick carbon foam heat shield capable of withstanding temperatures up to 1,800 degrees Fahrenheit. Detailed scientific data from this flyby is expected to be transmitted back to Earth later in January 2025. A recent study from the University of Arizona has unveiled a remarkable capability in certain patients with artificial hearts: the ability to regenerate heart muscle. This discovery could pave the way for innovative treatments for heart failure, a condition that affects around 7 million adults in the U.S. each year and accounts for 14% of all deaths. The research, co-led by Dr. Hesham Sadek and published in the journal Circulation, indicates that these patients regenerate muscle cells at a rate over 6 times higher than those with healthy hearts. This finding underscores the heart's intrinsic regenerative abilities, which may be enhanced by the unique conditions created by artificial hearts. Dr. Sadek suggests that artificial hearts may induce a resting state for heart muscle, facilitating recovery. He draws a parallel between muscle recovery in sports injuries and the potential for artificial hearts to boost heart muscle regeneration. However, he also notes that the heart's inability to rest could limit its regenerative capabilities, a concept he has been exploring since 2011. The research aims to determine why only about 25% of artificial heart patients show signs of muscle regeneration and how to enhance this response across all patients. This study is part of a broader initiative to improve heart failure treatments, which currently rely on heart transplants or devices like left ventricular assist devices, or LVADs. Utilizing tissue samples from patients with artificial hearts, the study involved collaboration with researchers from the University of Utah and the Karolinska Institute in Sweden and Germany. The implications of this research could revolutionize the management of heart disease, particularly as artificial hearts are already established as recognized medical devices. Dr. Matthias Magoola has made headlines by receiving the International Molecular Biologist Award for his impactful work in molecular biology and human health. His research primarily focuses on mRNA technology, a revolutionary approach in the production of vaccines and therapeutic drugs, positioning him as a key figure in this rapidly evolving field. Dr. Magoola notes that the successful validation of mRNA technology through the development of COVID-19 vaccines has streamlined regulatory processes and significantly lowered healthcare costs. His findings indicate that mRNA technology can produce therapeutic proteins more affordably, a breakthrough that could benefit billions globally. He is particularly concerned with the high costs associated with therapeutic drugs and vaccines, which affect over 6.5 billion people worldwide. To address this, he advocates for increased drug production capabilities in developing countries. Under his leadership, DEI Biopharma Limited has achieved notable success, including the development of a patented anti-malarial drug recognized for its effectiveness. Dr. Magoola is also advancing mRNA treatments for serious conditions such as acute kidney failure and cancer, receiving positive feedback from the FDA. In addition to these initiatives, he is working on vaccines targeting neurodegenerative disorders, including Alzheimer's disease, with anticipated FDA approvals on the horizon. DEI Biopharma Limited is making significant strides in Uganda, establishing the country's first facility for manufacturing biological drugs and vaccines in the Wakiso district. Dr. Magoola holds a PhD from the European International University in France and has published extensively on drug interventions for Alzheimer's disease, showcasing a broad range of research capabilities. His award-winning study, published in the International Journal of Molecular Sciences, highlights the humanitarian potential of mRNA technology as a cost-effective alternative to traditional recombinant methods. His achievements have garnered multiple recognitions, including membership in the elite scientific society, Sigma Xi, and being named a pharmaceutical CEO to watch in the year 2025 by the Silicon Valley Review. Researchers at the University of Pittsburgh have developed a new nasal swab test that can diagnose specific subtypes of asthma in children. This innovation could significantly change how asthma is understood and treated. The study included over 450 children and teens from Puerto Rico and Pittsburgh, focusing on populations that experience notably higher asthma rates, particularly Puerto Rican and non-Hispanic Black participants. Senior author Juan Celedon emphasized the urgent need for new therapies, as asthma remains the most common chronic disease among children, with a disproportionate impact on Black and Puerto Rican communities. Results indicated that 23% to 29% of participants had T2 high asthma, while 35% to 47% had T17 high, and 30% to 38% were identified with low endotypes. Notably, T2 low asthma, linked to air pollution, was found to be the most prevalent subtype among the participants. This test aims to better align asthma patients with suitable treatments, particularly for those in high-prevalence groups facing higher mortality rates compared to their white counterparts. While biologics are available for treating severe T2 high asthma, no treatments currently target T17 high or low endotypes directly, revealing a significant gap in asthma management. The new technology must receive FDA approval before clinical use, and further research is needed to evaluate the consistency of asthma types in children over time. The findings underscore the need for personalized treatment approaches, as existing diagnostic methods primarily identify T2 high asthma, often overlooking other subtypes. This nasal swab test could facilitate research into lesser-studied asthma endotypes and potentially lead to new biologic treatments, broadening the options available for managing asthma. These findings were published on January 2, 2025 in JAMA, arising from 3 independent studies focusing on Puerto Rican and African American youths who face higher asthma rates and mortality. Experts like Gustavo Matute-Bello from the NIH have noted that enhanced testing tools for biological pathways in children's asthma can significantly improve outcomes, particularly in minority communities. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. We'll be back Monday with everything you need to know.